All right, good morning, everyone. Welcome back to Plant Science and Technology. I'm going to try to make this recording and see how this works out. So just remember that midterm exam is due tonight, 11:59 p.m., so please log on and complete that. Otherwise, we're going to get into Chapter 9, one of my favorite chapters here, talking about genetics and propagation. So bear with me as I share my screen here. All right, so we're looking into how plants, the genetics of plants, are incredibly important for crop production, and as well as getting into how we make new plants, how we propagate them both sexually and asexually. So to start off with some basic genetic concepts in plant science, we have to think about where the plant or crop in question came from. So all the plants that we cultivate originated from wild ancestors, but most of them appear very different than their wild relatives. And these differences have started kind of based on just this recombination of genetics, as well as the redistribution of their heritable traits through the generations. So I think it's important to look at the origins of plants, where our crops came from. So this chart is just showing different crops from around the world, and there's a key there at the bottom of where these plants originated. These centers of origin of these plants are also where their wild relatives still exist, and therefore where the gene pools of those plants are. So we talked about early on about Nikolai Vavilov and how he kind of created these centers of origin, postulated the theories about where these plants came from. And so we can see some of the things here in North America. We have squash, strawberries, some grapes, sunflowers, blueberries, cranberries. It all originated on our continent. And if you look at where these plants are in the wild, that's where their widest gene pool exists. In Central America, we have things like maize, corn, other squashes, beans, peppers. In South America, we have other peppers, tomatoes, potatoes, as well as tropical fruits. So all these plants, originated different places around the world. Throughout our travels as a species, we've moved these plants around the planet, and now they've become part of our globalized trade. So the important thing, though, is just to note that where these plants came from exist these very diverse gene pools that often plant breeders look at for getting particular genetic traits and incorporating them into the gene pools of our crops. So we do this through chromosomes. Chromosomes are kind of where the genetic information is stored in the cell, in the nucleus. Most plants consist entirely of living cells, others made up of both living and dead cells. In the living cell, we have the cell wall, the cytoplasm, and the nucleus. The nucleus contains the chromosomes, where they exist in both complete form as well as in sex cells in the haploid form, where there's just half of the set of chromosomes, which recombine when plants undergo meiosis. Well, all organisms undergo meiosis in the sex cells of plants. So for us in the plant world, we're talking about the ovules, where fruits and seeds are created, or in the haploid, that's a diploid, having both sets of chromosomes where they recombine. The haploid organisms or cells are the pollen and the ovules themselves. So when they recombine, they get a complete set of chromosomes, half from each parent. And the number of chromosomes is going to be the same in all the vegetative cells of a plant species. One of the things that makes an actual species a plant. So if we look at the tomato, Solanum lycopersicon, they're all going to have the same number of 23 chromosomes in their vegetative cells. In their haploid cells, though, they have 16 chromosomes. So when they recombine, they get the full set. So we have 2N or diploid as a regular chromosome number, and then in the sex cells, it is reduced by half, called haploid. So the number, size, and appearance of these chromosomes are going to vary considerably between plants of different species, even in the same genus. So if we look at Solanum pimpinellifolium, which is a different species of tomato, it could have the same number of chromosomes, or it could have a different number of chromosomes. And in the genetic world, we've made strides in this science over the past few decades. And now the chromosome numbers and the genomes are known for most plant species. Chromosomes themselves have this great surface area. They're really long thread-like structures that contain the DNA, the deoxyribonucleic acid, and their associated RNA or ribonucleic acids, as well as some proteins that translate those. DNA is the active genetic material that can replicate itself and transmit different information to other parts of the cell, which code for how the plant grows and what it looks like. And it is a polymer, very large molecule made up of many of these repeating units. We discussed last week the nucleotides, which vary. So here's just an image of a DNA strand. And the backbone, as we can see here, is composed of different sugar residues, here in S, linked by phosphates. And so you can see how that alternates between the sugars that create the hydrogen bonds where the nucleotides are corresponding, bonded together with this phosphate on each strand. The bases that we have here in the center, the nucleotides, are adenine, A, thymine, T, guanine, G, and cytosine, C. And they are held together by hydrogen bonds that are attached to the sugars on each strand. So during cell division, whether it is mitosis, which is just kind of asexual, kind of recreating a cell, replicating itself, or in meiosis, where they're creating the sex cells, during that cell division, the chromosomes split apart, and the DNA molecule replicates itself by unraveling and separating at the hydrogen bonds, and then reforming into a new double strand. So DNA is the self-perpetuating genetic code, but it's the RNA, the ribonucleic acid, that actually controls the cell growth. So the DNA is double-stranded, whereas the RNA is just one single strand. And the RNA sugars basically have a place where they are translating the DNA for it to recombine. So the DNA acts as the template, then the complementary strand of RNA is formed. And then the proteins are constructed from the different amino acids using that RNA as a template, and then rearranging it in this specific arrangement determined by the bases of the original DNA. When we get down to the very base form of it, we are looking at genes. And structurally, a gene is just a sequence of 3 organic bases along a DNA molecule. The gene is the ultimate hereditary unit as part of the chromosome, determining the development of a particular characteristic. And we can't see this even with the use of an electron microscope. One gene or several interacting genes in most cases can actually determine how high the plant grows, the shape of the leaf is, color of the flower, or the size of the fruit. And any individual gene can have a large or small effect depending on what it is coding for. Some of those are going to act on their own to code something, while other genes act with others together to code for something. We also say that the genes that are on the same chromosome are linked, and they move from one cell generation to the next as a unit. Linkage, though, is not perfect, and often during meiosis, during that sexual recombination, chromosomes can break apart and exchange parts. And this is where we get recombination of DNA, basically creating differences in plants. In the vegetative cells, we have homologous chromosomes because they have the same gene or genes affecting the same traits at corresponding positions. Genes are also termed alleles. You probably heard this term before when you're working on the Punnett squares, as how the gene is expressed. So if they occupy the same position on a homologous chromosome and they affect the same trait, then they're linked. And they can be the same or they can be different. Alleles can also be seen as dominant or recessive. Dominant meaning that it's going to dominate, and even though you might have heterogeneity there, that one of them is controlling the expression, whereas the recessive is just kind of the hidden gene. So the dominant gene causes the characteristic to be expressed, whether it is homozygous, meaning it has both dominant alleles, or heterozygous, meaning that it has one dominant and one recessive. Homozygous could also be two recessive alleles. That recessive gene causes the characteristic that we're looking at to only be expressed if both of those alleles are recessive, though. So for instance, if we look at humans, and look at eye color, we've got, say, blue eyes, for instance, versus brown eyes. And brown eyes tend to be a dominant allele. If you were homozygous, one person is homozygous for brown eyes, their partner is homozygous for blue eyes, meaning one has both dominant traits for brown, the other has both recessive traits for blue. When they, cross, and when those chromosomes recombine, then the following generation, their children are going to have brown eyes because it's a dominant allele. But then they will be heterozygous for that gene because they'll still have the recessive allele in there. Then if they were to breed with someone who also had the heterozygous characteristic, there would be a chance that some of those children would have blue eyes because both of those alleles are in there, if that makes sense. So regular cell division in vegetative cells is mitosis, just the replication of the cell itself, basically cloning itself. And basically one of the chromosomes are still splitting, but they're recombining the same way. We can get mutations, though, that occur in mitosis. That's why sometimes you might see one leaf that looks different from the others, because that replication is not always perfect. Meiosis, on the other hand, which is also referred to as reduction division, occurs in the sex cells of plants. And again, it forms the cells with which we're looking at the pollen grains or the ovules, the embryo sac that contains the egg. And half of the original chromosomes are going to come from each parent. And you can kind of see here where they are, the chromosomes are splitting apart and recombining to form a new generation, new set of genes. The maternal and paternal chromosomes are going to line up across from each other in the cell during meiosis, and then recombine with each of those haploid cells, creating that mixture. So we got one male gamete from one pollen grain uniting with the female gamete, the egg, to form the zygote. So we have two 1N, two haploid combining to create the diploid, the full set of chromosomes, which is going to develop into the embryo and then eventually into a new plant. It's going to have some traits from each parent. When flowering plants are fertilized, and we also have two polar nuclei that form the food storage tissue in the seed. And that endosperm, as well as the cotyledons, can serve as this, basically food source for the developing embryo. And in some cases, it's important to note that sometimes we can actually have these gametes existing in some of that material as well. So in the cytoplasm of the cells, we can get some genetic recombination there as well, not just in the sex cells. So the manner in which gametes, the sex cells, can segregate and reunite to form new combinations are looked at below in the Punnett squares. And in this case, we're looking at kind of like what Mendel did, basically. He looked at characteristics of peas, and he looked at those that had very similar characteristics that only differed in one trait. So in this case, we're looking at a tall pea plant versus a dwarf pea plant, excuse me. And in this case, it's a monohybrid cross because it's only differing in that one characteristic. In peas, we look at tallness as being a dominant allele over the smaller size. So when they recombine, they're passing off a tall gamete and a small gamete. So the F1 generation here is going to become a hybrid containing some characteristics of both the tall and the small, but tall is going to be dominant, so the plant would look, that generation would look tall. However, we do get, when we redo that cross in the second generation, because now we have heterozygous traits here, we have both the dominant and recessive alleles. When they recombine, we're going to get 3 that are tall because they have the dominant tall trait, and then 1 that is recessive, getting 1 dwarf plant. And this is where we get segregation occurring in that F2 generation to produce 3 different genotypes and 2 different phenotypes. So genotype being the actual genetic expression, tall dominant homozygous, tall dominant heterozygous, and dwarf recessive homozygous. But they're going to be expressed by the environment there as just being tall or dwarf. So this is just looking at inheritance. And in this case, we're looking at a peach, and we're looking at a dihybrid cross. And basically, the thing here is it just starts to get more and more complex as more characteristics differ from the different parents. So in this case, we're looking at fuzzy skin and smooth skin and different flesh color. So yellow versus white flesh color. And as you can see here, that whole Punnett square there, the kind of looking at trying to predict how those genes are going to express. In the dihybrid cross, it becomes more complicated, and we're going to get different traits altogether. So the most simple cross, and when we're actually breeding, we want to look at fewer characteristics, because when we have multiple characteristics, we have multiple genes interacting, which means that it's more difficult to control and predict how it's actually going to be passed along. As the plants grow, though, we have hundreds of thousands of cells constantly dividing, and that DNA replication is not perfect. So we have errors occurring, and this can kind of form as mutations, causing changes in the plant's characteristics. This is actually a way that plants evolve to their environments and how we see new characteristics appear. Unfortunately, the majority of mutations have a negative effect on plants. However, some of them are not and do aid in developing new cultivated varieties. Mutation rates can increase, are likely to increase, with the treatment of using, say, different chemicals, or actually, irradiating the plants with some form of radiation. These create hereditary modifications that can also cause changes in the chromosome number or structure, which can lead to making sterile organisms. The alteration of the genes themselves can express and alter characteristics of the new plants. Mutations, though, are not always passed along. Sometimes they just appear and then disappear. We only see it if we asexually propagate the plants. Another phenomenon that we see, especially in plants, is known as polyploidy. And it's basically a condition which individual plants have more than 2 sets of chromosomes in their vegetative cells. This usually arises as a result of the duplication of the chromosome sets from a single species, which is autoploidy, or it can also happen from alloploidy, which is a combination of sets from 2 or more species. However, many of our cultivated crop species actually evolved in nature as polyploids. So if we look here at a couple of different members of the oat genus, Avena, the sand oats and the slender wild oats, both wild versions of that plant, they have different somatic chromosome numbers. The cultivated oats, Avena sativa, then actually recombine from that somatic chromosome number and end up having 42 chromosomes. That basically gives a wider variety of genes and traits that can express differently. We actually see the same thing in triticum, in the wheat ancestors, einkorn and emmer wheat. Same deal. They have different somatic chromosome numbers, and then when they recombined to form the common wheat that we grow as a crop today, that had those chromosomes combined. So kind of just by chance, it's not something that we can easily induce. We've tried to make this happen through using different chemicals and different forms of radiation. But it's likely that in nature, this just occurred as a mutation, and that someone was there to witness that mutation and then cultivated it. Pretty neat. As I mentioned before, we can also get certain characteristics obtained through the cytoplasm in vegetative cells. On the genotype, we're looking at the genetic makeup of the plant again. And we're looking at the order of which the genes are situated, how they're linked, and how that makes the genetic makeup of the plant. The phenotype, on the other hand, refers to the plant's actual appearance, and that's going to be expressed as a result of that genetic encoding as well as the interaction with the environment. So the important thing to note here is that it's not just the genetics that we're seeing in a plant. We're seeing genetics plus environment. So phenotype is what we see, genotype is the actual genetic makeup. The environment can actually turn on and off particular genes. So for instance, if it's cooler, that plant is experiencing cooler temperatures, that can cause certain genes to turn on or off. When we talk about actual breeding of plants, the big thing that we are looking at is selecting different parents with which the desirable traits that we want are going to be passed on. And we can either just select them and let them reproduce naturally, or more often we will actually intentionally cross them together to produce new offspring with both sets of desirable traits. And plant technology, the science of technology, has developed incredibly in the world of genetics. So in the past, breeding programs could take years or even decades to develop a cultivar with the new desired traits that we're looking at, because we had to grow the plants to maturity and evaluate the characteristics. Now we can do DNA analysis even in very young plants and even in embryos to determine if those desired traits are present. And we can even actually do some breeding on a molecular level. This is kind of more an image of traditional breeding right here, where we're looking at cross-pollination. In this case, in dianthus, carnation flowers. And you can see that we've got a flower here with the male parts that we're removing and then placing onto the female part on the pistil. So we're actually intentionally selecting two different parents, moving the sex cells to recreate that fertilization and ideally to pass those genetic traits on how we want them, whether it's a larger flower, maybe a different color of a flower. The thing with breeding is it requires very high attention to detail. And the female must be kept from receiving unwanted pollen. So we generally look at trying to do some control of that. So generally what we'll do is use like male fertile plants to produce hybrid seed on self-pollinating plants. By doing that, we'll have to actually remove the male parts before that flower matures. So we'll go in there before the flower even opens, take out the pollen producing parts of the plant, and then reintroduce our own pollen via that hand pollination that we were just looking at. As I mentioned before, we're looking at the monohybrid crosses ideally, where we're just looking at single differing traits. Those are the easiest to breed for. The more genes, the more difficult it is to get those desired traits. It is possible to develop a homozygous line from one single self-pollinating plant, though, because when a population becomes completely or nearly homozygous, reduced vigor can occur. So we have to figure out different ways to, increase that vigor. And one way is just by hybridizing plants. And, hybridization is fairly new in the grand scheme of things. Genetics is too. It's a new science, and it's based on research that Gregor Mendel did in a monastery, scientific monastery, and he found that when we cross things that had totally different characteristics, then they produce an F1 generation that are going to have uniform plants because they're going to have very similar or the same genotype. They're homozygous, but hybridization causes the phenomenon hybrid vigor, where they're likely to outperform in many of the characteristics in either of the parents. And I mean, you guys are probably taller than your parents, right? You have different characteristics because of that hybrid vigor. You know, two people totally different, they recombine, you get that vigor, and that's often seen as a greater height. In the US, we're looking at corn. Almost all corn is now produced from hybrid seed, which just doing that has actually more than doubled yields around the country, around the world. Unlike the open pollinated crops, seed from hybrid crop cannot be saved and used to produce future crops. Well, it can be saved and used, but the F2 generation segregates. So we have different genotypes and phenotypes, so it's not reliable to save pure seed. We also have the great advances in biotechnology in recent years. And what we're looking at here is looking at the molecular biology and using things like genetic engineering techniques to create new mutations, new traits. And improvements in this process have enabled scientists to introduce plants that have foreign DNA. And most of what we see is in the more agricultural, the agronomic crops. So things like corn and soybeans, wheat, sugar beets, these things that are planted over hundreds and thousands, if not millions of acres. And most of the things that we're looking at for genetic engineering include things like enhanced dietary attributes, where we've actually bred rice, for instance, to have vitamin K with it, genetically engineered it. We've also genetically engineered crops that have resistance to insects or herbicides, or even certain environmental stresses. When we introduce a gene to a species that does not have that gene, it is called genetic engineering. And those plants, those resulting plants, would be called transgenic plants because they have a gene from different parents. And often different species, in many cases from totally different organisms. We use the term GMO kind of in the popular culture, genetically modified organism, but really the more accurate way to refer to it is genetically engineered plants, because technically we've been modifying genes for a long time just in selective breeding, whereas engineering is actually done in the lab. So a genetically engineered crop is different from, kind of the term GMO, in that a genetically modified organism is created each time sexual reproduction occurs because the genes are modified. However, not all genetically engineered plants are the result of gene transfer. Sometimes we're just altering the genes that are already in the plant. One of the classic examples of that was the flavor saver tomato. This is one that they were trying to take genetics from that existed in the tomato genome, in that gene pool, and make them, easier or more resistant to, shelf life, basically. Unfortunately, though, that flavor saver tomato tasted awful, tasted like cardboard, so they gave up on it. Genetic engineering can complement, but it cannot replace traditional breeding techniques. Just like breeding, genetic engineering would start with the identification of a gene that is going to give a desired trait. And then that gene is transferred from the host organism into the nucleus of the new plant, basically. We also have this neat technology that has been developed to actually carry the gene on a bacteria that basically goes in and recodes the genetics. And we can follow that by inserting a marker gene. It doesn't actually do anything for the plant necessarily, but is basically used to follow and see how that gene is being moved throughout the plant. We could use this in combination with traditional breeding to watch for particular genes. They're doing that at the American Chestnut Foundation now, if they can try to follow the resistance of the Chinese chestnut to chestnut blight, and see if they can get that gene into the American chestnut. So here's just a kind of simplified look at the steps of genetic engineering. We look at a flowering plant that has a gene that produces disease resistance. We find that gene, identify it on the chromosome, and then we actually use a type of bacteria to actually cut that snippet of DNA and then attach a marker gene to it to allow it to be identified, and then transform the cells and then reintroduce that. So in this case, they're looking at using this agrobacterium as a way to insert that gene into the nucleus, recoding that DNA into that plant cell. So now that it's in there, we'll actually try to transform the cells. In this case, we're looking at doing this in sterile media, growing it in a lab, to make sure that it's testing for that disease resistance. And then only the cells that contain the new DNA are going to survive and develop into new plants. And then those new plants are grown and reproduced through some sort of asexual propagation, cloning them to get many new plants and replant them. So only existing genes currently have been modified or transferred. We don't have the technological capability to create new genes. But we are able to insert those into cells and also use a promoter gene to keep it turned on, basically, under specific conditions. So today, it's going to be more than this. It's a little bit outdated, but even by 2008, 86% of cotton, 80% of corn, and 92% of the soybeans grown in the U.S. were genetically engineered to be resistant to herbicides or insects. And of course, the use of genetically engineered plants has greatly increased. And there's a lot of controversy about that, about what effects could that potentially have to our food supply. Generally, if you're shopping in the produce section at your grocery store, though, you're not going to see anything that has been genetically engineered. What we often see, though, is these products making their way into the food supply through processed foods. So you buy like a box of Chef Boyardee pasta. It's likely to have genetically engineered wheat in it because it was produced on a large scale. We don't really have any genetically engineered produce, though. All right, so we're gonna get into, especially on Wednesday, talking about just propagating plants, which is just this process of increasing the number of plants. And we're gonna look at how we can do that through sexual and asexual methods. Sexual methods, of course, we're looking at growing them from seed. In some cases, we might be able to do some in the lab sexual propagation as well, not necessarily from seed, but it involves that genetic recombination. Asexual methods, we're looking at just cloning the plants. So successful propagation transmits the desirable characteristics from the original plant to the progeny. And if the characteristics of the original are lost or changed, then that particular method is unsatisfactory for that type of plant. We have lots of different plants out there that all started as one individual. So right here, just some examples of that, that started maybe as a mutation, and then we cloned those plants. And so they've all come from one you know, genetically distinct plant and have just been cloned over and over again. Pretty fascinating. We also look at the open pollination, the sexual propagation of plants, as breeding true to type by seeds. If no characteristics are changed, then the cultivar is called a line. The line is homozygous, and if self-pollinated, it's going to yield progeny that's just like the original plant. And most of the world's leading economic plants, the grains, many of our vegetables and garden flowers are made up of these lines. Many forest species, although they're seed propagated, are not considered lines because of their higher level of variability. So we also have other types of seed propagated cultivars, such as the inbred lines used to produce hybrid cultivars, and then of course the hybrids, where we're basically keeping each line intact and reproducing it so that we can replant them to produce the same hybrid seed with the same success. Most of our plant groups are heterozygous, though, meaning that they have, multiple expressions of genes rather than homozygous. So there are many dissimilar genes controlling their characteristics. So in that case, we use vegetative propagation to clone them, where we're placing that vegetative tissue into a suitable environment and forming new plants. When we're using that type of propagation, we can perpetuate that heterozygosity without losing the characteristics. So we're cloning in that case. And we maintain those lines through continually cloning them. Basically, we're looking at a part of tissue that is just doing that mitosis, just regenerating those cells. We can look at that through a wide variety of asexual propagation, which we shall discuss, from cuttings to grafting, budding, layering, and also using runners. If we're trying to maintain a cultivated variety by seed propagation, we have to look at the seed source. If we have a homozygous cultivar that cross-pollinates with other cultivars or other species, then the plants that are used to produce those seeds have to be separated from other plants to prevent contamination and the loss of genetic purity. With certain plants that are propagated by seed, some of that variability might be tolerated or even wanted, especially if we're actually trying to adapt the plants to different conditions. Most U.S. states and many other countries also have established some sort of seed certification program to maintain genetic purity of cultivated varieties. So we have government agencies that are setting the standards for seed quality and different characteristics, which causes that certified seed usually to cost more. But the grower, the end grower that's growing that crop, is going to have assurance of better quality of the seed. And in North America, of course, we've become accustomed to using that certified seed from recognized producers. So here are just a few different classes of programs of U.S. seed certification. We look at breeder seed, which is produced in small amounts, mostly just for the genetics, for breeding stock for breeders to use. Foundation seed is that is multiplied from breeder seed. It's available in limited amounts and often controlled by public or private foundations. Registered seed is what usually goes out for growers of certified seed. And then certified seed is the most simple, what is often sold to the end grower, the farmer, is just for general crop production. It's of a known genetic identity and purity and is available in large quantities. Real quick, we're going to just look at seed formation again and how the seed has three essential parts. Living part, the embryo, the food storage material that nourishes it, and then the coverings of the seed. For a seed to germinate, of course, that embryo does have to be alive. Makes sense, right? Some seeds are very short-lived and are only viable for a few days or months, such as a lot of deciduous trees like willow, maple, and elm. Others can stay alive for many years, like the hard-seeded legumes. That viability, though, is going to depend on the storage conditions. Generally, best if they're under refrigeration and at low relative humidity, so cooler and drier to retain viability. It is advisable to test the viability of a representative sample of the seed that's going to be planted. And there are a few different ways that we can do that. The most simple way is just to float seeds in water. And often the floater seeds, those that rise to the top, are empty, do not have a viable embryo. We can skim them off. The ones that sink do have a viable embryo and can be planted. We also use a germination test to see what the percentage is going to look like, basically by putting it in the proper moisture and temperature conditions in a controlled environment and just counting the number of seeds that germinate. There's also a tetrazolium test, which is using this chemical that turns red when it contacts living and respiring tissue. So they usually soak the seeds and then place them in a tetrazolium solution. And if it turns red, then the seeds are viable. There's also an excised embryo test where the embryo is removed from the seed, placed on moist paper to see if it will show some activity. So this is all looking at dormancy. And dormancy is a very important survival mechanism for many species of plants and is a result of evolutionary development, which allows, many plants to go dormant over autumn and allows them to, then come back and grow the next season. So we have a couple of different types of dormancy. We've got seed coat dormancy, where basically the seed coat is preventing, moisture or other gases from penetrating the embryo and initiating germination. And we often have to soften that seed coat in order to overcome this dormancy. We can either do that through scarification, where we're actually mechanically altering the seed coat through things like sandpaper or soaking in an acid or heat treatment, often through like boiling water. We also have embryo dormancy, where basically that embryo is not going to grow, and even though it's got the right environmental conditions. And generally, we see this in a lot of our native plants in temperate areas. So we get some tree seed. If we don't, allow them to go through a process that is similar to winter, then that embryo will not continue to develop. So that's stratification, where we're keeping them in cool, moist conditions in order to get them to kind of go through that wintering. We've got to look at the temperature, the moisture, the amount of oxygen, and the time for that. So often we'll stick seeds in like some moist media and put them in a freezer or a refrigerator for stratification. Stratification actually does increase growth promoting hormones and decreases growth inhibiting hormones in order to allow those plants to break through that dormancy. And then some species have seeds that have both seed coat and embryo dormancy, and this is known as double dormancy, If you want to get them to germinate, you have to treat them and you have to scarify them and stratify them, basically. So here's just an image of epigeous germination, where the plant, the cotyledons are pushed above the ground with the seed still attached, or where, in this case, in a peach tree, where the cotyledons will remain below ground and the true leaves come out. That's called hypogeous germination. Here's what happens when we get seeds to germinate. First, we have the imbibition of water, which water is let in through the seed coat and allows the cells to become turgid, the cell walls to develop. Then we have the activation of hormones and enzymes after that water is absorbed, where these enzymes are activated or synthesized, often as a result of that stimulation of those growth hormones. And the food materials are moved from that endosperm to the growing points of the plants. Then the embryo continues to develop in both directions through cell division and enlargement. So here we have first the radicle emerging, and then we have, once that seed coat has been broken, the radicle emerging, and then the actual plant developing and creating leaves to create its own source of food. Last thing here, just some of the environmental factors influencing germination. First, we have to have moisture. We've got to have water in order to initiate those processes. We've got to have the right temperature, which is going to vary with the type of seed. We have to have some aeration. Generally, we need some oxygen there. So a media that is able to provide the proper moisture and oxygen is very important. In some cases, some seed need light to germinate. Others need a complete lack of light. And finally, we also need the freedom from pathogenic organisms. So Pythium, Rhizoctonia, and Botrytis are all different types of bacteria and fungi that can cause this damping off syndrome, which basically just causes the developing seedling to rot and just completely fall over. Also, we have to have freedom from toxic amounts of salts in the soil, which would just dehydrate the growing seedling. All right. If you guys have any questions, comments, or concerns about this, please just let me know as soon as possible. Look forward to seeing you all on Wednesday. Good luck on that exam.